I'm super pleased to introduce Suzanne Anker. Both of us flew in from New York last night. And it's really a recovering city. It's really tough, actually, what's going on in New York City. But we're back here, sunny California. And Suzanne will talk about her work. I will introduce her briefly with her amazing biography and the amount of wealth that she can share. She, to me, is very much an example because she is a practicing artist who exhibits, has always exhibited in the fine arts world, has always experimented, crossed over in many different fields, but at the same time expanded out by publishing, by lecturing, teaching, and since 2005 she has been the program director of School of Visual Arts of the Fine Arts program, which is best school, one of the best schools in the arts in New York. So not a small thing, a big responsibility, but she's never stopped working as a fine artist. And this is what's very inspiring to me. So I'll just give her a few accolades. I have to actually read because it's so much, but I'll just skim through it and give you the kind of highlights. So her work has been shown in Walker Art Center, Smithsonian Institute, the Phillips Collection, PS1 Museum, J.P. Getty Museum, Central for Cultural Inquiry in Berlin, the Para Museum in Istanbul, Museum of Modern Art in Japan, etc., etc. Her book, The Molecular Gaze, Art in the Genetic Age, is a textbook for me and for my students, and she's really pursued to looking into the meaning of genetic age, the molecular gaze, truly, as an artist, also as a historian. So it's a highly recommended book. She's also published Visual Culture and Bioscience, and she writes and publishes her essays in journals like Seed, Art Journal, Tema Celeste, all of these, New York Times, any place and every place where this subject is addressed, eventually you'll find Suzanne there. She also hosted, and you can find this online archived, about 20 episodes on the online art radio that was in collaboration with the Museum of Modern Art in New York. It was called Art on Air. So just to end with the accolades, I'll say that she's been a speaker at the Royal Society in London. She just spoke at Harvard, which we're very impressed with. Yale University, the London School of Economics, Max Planck Institute, and still does her artwork, so we love that. Now, what I'm particularly impressed with is that she has basically set up the first biolab in a fine arts department. And I went and did a little interview. It's 4 minutes. I want to show you because you'll actually see it. So here we are. Sound? Here we are at the first biolab in a fine arts department, following the tradition of beauty of natural anatomy, of nature in general, what artists have always been obsessed with, we have it here. So what you see in biotech labs, now you're seeing in the fine arts department. Let's go see what Suzanne can tell us about it. Okay, so where do we start?
Why don't we start over here and take a look at our fish. And we have fish and shrimp and frogs, and we are just sort of beginning to develop what it means to create an ecosystem. So hopefully, once these are more established, we will have students actually given one of these terrariums to create a particular kind of
So
ecosystem.
is one of your goals to emphasize the environment
Yes.
with the arts?
Yes, I think one of our goals is really to integrate visual thinking into living, and the ways in which we are responsible and personally responsible for all aspects of the living world.
Wonderful.
So, another aspect of this has to do with what Orrin Katz calls the aesthetics of care, in the sense that if you're going to work with live material, then you have to come back on a regular basis in order to keep it thriving.
That's right. That's right. And then here
We have more of our skeleton collection.
it's funny because I think what I got very attracted to when I saw this, and I just thought, wow, it's like a full circle. I studied in a very classic fine arts school in Eastern Europe, in Belgrade. And we did bone by bone. And I used to think, why am I doing this? This has nothing to do with reality. But it kept coming back.
That's right.
So the knowledge of this,
That's
I
right.
thought was so important, but I could not convince art faculty or students. They always thought, what is she talking about? So to see it coming back into the fine arts
Absolutely.
is so wonderful, so
And-
important.
And we have several courses in this. We have an
Fantastic.
human anatomy course, and now we have a comparative anatomy course because we have the skeletons of many different animals.
Yeah.
So, you can. Well, let me tell you the story behind these because these were a big surprise. I am working on a series of pieces in which I am converting sea sponges into porcelain, which you have seen. And part of the process involves soaking the sponges and kind of going through them to make sure that there's no matter that would disturb the kiln once we put it in that high temperature. And all of these things have been excavated
Amazing.
out of the sponges. So there's different kinds of symbiotic, parasitic relationships that range from different kinds of little shells, mollusks,
All in the sponge.
all in the sponge,
Whole
to
universe.
starfish,
Whole community.
community,
These
to
guys
hermit
too.
crabs,
Oh
yep,
my goodness.
to coral, different, and these of course are the skeleton of the coral. The coral was once alive. So this was a surprise to all of us, and we'll probably consider this kind of excavation or field work as a project in the future.
Very nice. So please give a warm welcome to Suzanne Anker.
Well, thank you, Victoria and UCLA and everyone who's here tonight. I'm thrilled. I haven't been in Los Angeles for a very long time, and I must say it's quite beautiful to see all the flowers in bloom. Today I'm going to talk about the genetic seed bank in terms of a form of recovery and reclamation. But before I start that, I just want to say a few words about the role of technology and its impact on our lives, and how its ascendancy is literally plugging us into the world. But in this ascendancy, what we're finding is that everything, pictures and data and sculpture, essentially is created by these zeros and ones. And this is very important in terms of the ways in which pictorial wars are essentially fought. And these digital images circumnavigate the globe, and I like to refer to them as signs in action. The other part of that question is the way in which compelling narratives are embedded in these pictorial presences. And since we are dependent, particularly in a media-driven society, on the notion of symbolic pictorialization, the question remains is how these representations operate as a mutating cultural imaginary. We can consider the term cultural imaginary as an overarching term defined as those vast networks interlinking discursive themes, images, motifs, and narrative forms that are publicly available at any given culture at any one time. And what they do is articulate its psychic and social dimensions. This is what states the cultural critic Graham Dawson when he talks about this term, the cultural imaginary. It constitutes a semblance of a collective database, and it traverses contested territories associated with either verifiable axioms or fanciful storytelling. These are visualizing models. They're employed by artists, scientists, designers, corporate advisors, journalists, politicians, and they do a variety of things. They clarify, they mislead, they aggrandize, they stimulate, and they even document. In short, they are representations embedded in social structures, as well as policy decisions and commercial ventures. Question that is never asked in fine arts, and which may actually be a necessary question to ask, is what values are embedded in a particular work of art? So when we think about extreme cases of performance art, for example, what in fact are we looking at? And what these images do, or all aesthetic devices do, is they perform a semiotic function of activating thought, as well as emotion through their powers of communication and circumscribed belief. Now, conventional discussions with regard to the visual arts in relation to science and technology has taken a spirited turn. Set in accelerating motion by the decoding of the human genome in 2000, many visual art exhibitions, residencies, and symposia, as well as art-sci collaborations, have been active throughout the world. And current discourses in visual arts are also expanding, in my opinion, beyond the nomenclature of formal analysis, Marxist and feminist interpretation, post-structural theory, and alike. If we add to the mix the concept that art is a form of knowledge production, then how can we address art objects in the 21st century? Now, art practices has become a set of shifting dialogues which are being explored in terms of several platforms for revisioning the world. And I'd like to mention a few of these to you, some of which you've heard, some of which maybe you haven't. The first one is art as speculative research, which is a platform in which art practice reaches beyond gallery or museum walls to develop interactive social and biological artworks. And these include sophisticated interactive technological apparatus, changes in working conditions and labor relations, which come about by changes in consumer demand for technological hardware. And an example of this is Lisa Ma, who had studied Reproductive Design at the Royal College of Art in London. And you can look at her work online, particularly a piece called Farmification. And what she did was go into the interior of China and to embed herself with a series of workers who made joysticks. Now, when was the last time you heard that as a technological device? And the workers there were originally farmers, and they were migrant workers. So whenever there was not enough demand in the first world for these technological devices, which were essentially expired, the migrant workers would have to go to another place. So she decided that there's all this land right around the factories, and what would it be like if she could purchase this land, which she did for about $2, and set up a situation in which, when there was not enough work at the factory for these defunct technological devices, that the migrant workers could do some farming, grow their own food, etc. And this worked out so well that many other factories in the neighborhood also started this practice. And she called this project Farmification. Another kind of shifting dialogue, which is being explored now in art as a form of practice-based research. And we see this a lot in the generation of PhDs in studio art, which may or may not be a good thing. The jury is out on some of those questions. But I think when one begins to look at what Linda Candy's Guide to Practice-Based Research cites, she says the following: the creative artifact as the basis of a contribution to new knowledge is one of the ways in which art practice, and I'll just sort of quote someone else, Reinberger says, becomes a generator of epistemic objects. So in a way, the object contains the knowledge that it took in that time and place to generate itself, and the object talks back. And the third platform that you may not have heard of is from a German philosopher named Nicole Karafyllis, and she has coined the term biofact. And those of you who've ever studied philosophy or are in the philosophy department know how impossible it is to create a new word. And what she did is she took the prefix from biology and the suffix from artifact and combined them to create these new entities that are being generated in the laboratory as I speak. And she says that the biofact is a hermeneutic concept which allows one to inquire into the differences between nature and technology in the domain of the living. And she argues that these differences do, in fact, still hold true today, as they did for Aristotle, but the distinctions have become much more hidden than before. So for Karafyllis, a critical point becomes the invisibility of what she terms the veil of ignorance associated with manipulated life forms. These transformed bodies, whether in the form of a GMO or a laboratory animal, conceal the changes in the DNA status. So where seeing was once believing, that too has been compromised. I'm going to turn our attention now to a series of projects of mine which intersect the world of visual arts with scientific concepts, and give you some clues into the kind of magical thinking that I myself and many other artists are engaged in when they repurpose signs into works of art. So I'm going to quote one more theorist who talks about the social field of images, and that's W.J.T. Mitchell, who likes to think about scientific images as increasingly entering the public domain. So it underscores for him the pictorial turn across disciplines, which of course is coupled by the lingua franca of digital media to create pictorial presence out of visual data. So let's begin here with some of these pieces. And I think that it is critically important to look at art as a critical fiction, whether or not it's part of the cultural imaginary or speculative design or some other kind of hybrid practice. I like to look towards literature, and particularly Margaret Atwood, and particularly her novel For Oryx and Crake, which is a dystopian novel about the future of mankind. And she opens with one of the main characters called Snowman, and this is what she says. Snowman wakes before dawn. He lies unmoving, listening to the tide coming in, wave after wave, sloshing over the various barricades, wish-wash, wish-wash, the rhythm of a heartbeat. He would like to believe he is still asleep. On the eastern horizon, there's a grayish haze, lit now with a rosy, deadly glow. Strange how that color seems tender. The offshore towers stand out in the dark silhouette against it, rising improbably out of the pink and pale blue of the lagoon. The strikes of the birds, that the nest out there, and the distant ocean grinding against the ersatz reefs of rusted car parts and jumbled bricks and assorted rubble sounds almost like holiday traffic. Out of habit, he looks at his watch. Stainless steel case, burnished aluminum band, still shiny, although it no longer works. He wears it now as his only talisman. A blank face is what it shows about him, 0 hour. It causes a jolt of terror to run through him, the absence of official time. Nobody, nowhere knows what time it is. And I think that in thinking about Atwood's work, I created a number of pieces by visiting scientific laboratories, mostly in Europe, in which I began to question the ways in which nature rebels. And by putting nature in sort of a little box and thinking that it's going to stay there is almost like the image we see here of the straw man who is looking at his DNA sequences. I did a series of pieces about this in which I superimposed gardens into the images of laboratories, not knowing whether the nature was returning or nature was leaving. So the kind of ambiguity of these conjunctions raised the issue of what is the new alien. And what we see here in laboratory life is actually a biophysics lab in which this reactor was the apparatus that could measure the electric charge in one neuron. But I'm not alone in this sort of question about nature and culture and the way in which they have folded in on top of each other. Certainly in the Hudson River School and in Romanticism, which is also concomitant with the rise of photography, we see that both instrumentalized vision and a kind of re-spiritualization of the natural world was ever present, that it's a kind of secularization using nature as the spirit itself. Thomas Cole, The Last of the Mohegans in 1826, of course he's a man of his time, and little did he really talk about the genocide that is present here. He is trying to portray America as the new Eden. And in 2000, if we kind of look at what this landscape is like, we see the atmospheric pollution in which chemical fallout is creating a very similar kind of rust-colored awe, but to negative effects. Even more startling is Frederick Church's work, Twilight in the Wilderness, in which we see this beautiful scene of a untouched landscape. When we look at the red sky these days, it represents pollution for us. Also, 1860, this is a time that the Civil War was going on. And Frederick Church is pretending that we are essentially locked in our beauty of spiritual desire. You can see it in the piece that it's compared to, that the same kind of glowing form is represented in this explosion at the nuclear power plant in 2011 in Japan. So what was once a sign of transcendentalism is no longer that. It now has other kinds of associations that talk about the way in which technology is even more fierce than one could have thought it was. And if I quote John Dewey in his seminal text, Artist's Experience, he talks about the experience of art as a civilizing process. And it's akin to developing moral judgment. That's why I kind of asked the question about works of art, what are the values they embed? And going beyond its material significance, art becomes a communicating vehicle in and of itself. Now, much of Dewey's moral philosophy was necessitated by the shifts in living styles generated by the accelerating demands of new urban life. Yet in developing moral judgments in a techno-scientific society, this is yet another task. And here you can kind of see in the Beardstadt the animals that are roaming free, whereas in our factory farms, we see the animals that are penned in. This pile of manure that we're looking at as well is more than manure itself. My mother always used to tell me, in a very gross way, never shit where you eat, okay? Now, she didn't say never eat shit, because this shit is mixed in with straw and hay as a food product for the cattle themselves, and it's called wastage, and it is there to cut costs. Which brings us to repair and recovery, hopefully. And one of the things that have interested me since the late 1980s is the way in which genetics has not only entered popular culture, but has become the umbrella for almost every aspect of our lives, from the food we eat, to the clothes we wear, to the medicines we take, etc. But the question here remains also is, what kind of language do we use when we talk about DNA? Do we say it's the blueprint of life? Do we say it's a family trust? Do we say it's a bioarchive? Do we say it's a deed to a body part farm? And do you own your own DNA? And the answer to that is no. So this is a whole other lecture in and of itself to essentially parse the way language is used to present the inside information about what some of the possible kinds of scenarios are when we manipulate DNA and alter life. In 2007, I was heading down to the Miami Basel Art Fair, and I thought, well, I want to do something even more interesting. So I went to visit the Moat Laboratory, which is a laboratory dedicated to the preservation of coral reefs. Now, coral reefs around the world have been so decimated that 90% of them are gone. And as one of the ways in which they can be restored is to begin to grow them indoors in tanks, which you can see here. And using industrialized kinds of pipes that are used sometimes for plumbing, PVC, what one finds is that little pieces of coral, almost looking like petit fours or little desserts, are positioned on these concrete slabs. And the scientists found that the concrete was a very good substance that allowed the coral to grow. Now, coral is a very complex creature. It is both a carnivore and one that houses a kind of symbiotic plant in which the coral can produce its own food using photosynthesis. And in the 19th century, coral was thought to be a plant. Some of these categories still exist today. So, for example, in many science labs, one would study mushrooms in a plant kingdom, even though they're not plants. They're somewhere between an animal and a plant. And looking at these corals in these CO2 chambers in Moat, and these will be exhibited, these pictures will be exhibited in the exhibition here in large-scale prints, we see that there are relationships between the coral form and the brain. And one may ask, why do we have brain coral that looks like the brain? Why is the brain shaped the way it is with these convoluted kinds of linear dimensions? And the reason for that is that there are many more connections that can be made between parts of forms if this kind of configuration is present. So it's no coincidence that our brains sort of have these convolutions, and it's no coincidence that coral, which function as a colony or set of colonies, would have these as well. Another tank with purple algae and the green coral, which has no skeletal structure. None of this is photoshopped, by the way. And an outdoor piece in which the coral is now adjusting to its environment. And what the scientists do with deep sea divers is they take these bits of coral and they place them back into spaces in the ocean that have not been overrun by detergent and shipwrecks and skiers and water skiers. And they are also forming the first kind of genetic analysis of different coral species and where they come from. which led me to my next piece, which sort of happened not in the context of Carl, but in the context of the brain. And in 2011, I was asked to curate a show about neuroscience and art in Istanbul at the Para Museum, and I began to look for another metaphor for the brain. How do you show the brain? How do you picture the brain? Yes, we can do the morphology of the brain. We can do somewhat the brain in action with fMRI. But what other metaphors are necessary or capable of really carrying this kind of thought? And which led to the next two questions. Why do we think in terms of metaphor? Why don't we just think this is this, as opposed to this is like that? And in looking through the literature, George Lakoff, a philosopher and linguist, talks about in his neural theory of metaphor that essentially metaphors take the form of cross-domain mapping. So they're able to put together disparate information the way many artists work, maybe even in a form of magical thinking, and they create an infinitely complex set of connections that we use when we perform our actions. More recently, also, the brain has been studied to show how plastic it is, meaning that it fires neurons under different conditions in both different and identical ways. And most recently, with mirror neurons, it was found that brains will fire the same neurons whether you're eating an ice cream cone or you're just dreaming about an ice cream cone. And this, of course, functions with phone sex, which is not really sex, but it has to do with the neuronal makeup of how we act in fictive situations. And back to the cultural imaginary, back to the importance of art as a way to see the world. And in sort of continuing with this piece, I began to study the sea sponge and found out that besides being surrogates for the brain, they also had other characteristics that interested me. Number one, we share 70% of our genetic material with the sea sponge. Now, we don't look like sponges or daffodils or any of these other creatures, but what's important about the sea sponge is that it's the first multicellular organism, and it has no nervous system. It has a proto-nervous system. So it is now being studied in the laboratory as a model organism to find cures for Parkinson's and ALS and other kinds of really horrific degenerative diseases. Why would an organism have the building blocks and never use them? If we could find that out, maybe we can create the building blocks and use them. And I don't mean me personally or you. I mean people who were involved in this kind of science. Another thing you'll see in the biota pieces are these little amulets that are attached to the porcelain sea sponges, and they represent little figurines that one would find in prehistoric societies. And again, in looking at the literature, and this is where art as a form of knowledge production or as a form of research enters into the equation, is that other theories have to do with the fact that a new, very old figurine was found in Germany that makes it look like perhaps the Neanderthals or the Homo sapiens at a certain time in culture actually experiences changes in the brain which was converted into the form of figurative sculpture. So the fact that conceptually speaking, we can project ourselves into this figurine. I'm using these figurines as kind of stand-ins for evolution and its ideas. I then came across this poem by Emily Dickinson, and this is what she says. She says, "The brain is wider than the sky, for put them side by side, the one the other will contain, with ease and you beside. The brain is deeper than the sea, for hold them blue to blue, the one the other will absorb as sponges buckets do." So sponges are a good metaphor for the brain in many ways, but here they look like coral. And when we go back to the blight and devastation of coral itself, these questions remain of what are we going to do about this to heal not only the planet, but ourselves. All right, which brings me sort of to my next idea, which is about reproductive technologies, which are ironically called ART, Assisted Reproductive Technologies, ART. And if we look at the literature again from 1954 and 1965 and 1958, we are going to see how technology is embedded in the war machine and the industrial complex, and how the same kind of language used by Life magazine is being used today. So if we look at the piece on the left, you'll see the sound wave portrait in the flesh. And this was a somascope in which this company could now use a device, a rather large-scale device, in order to somehow assess soft tissue tumors. In 1958, what happens is that this somascope is being used. A woman comes in to see her doctor complaining of a stomach ache. He gives her this somascope kind of test and finds out she's pregnant with twins. And this is the origin of the sonogram. Now, the reason we don't have the sonogram before 1954 or 1945 is because sonar rays and sonography was classified because of World War II. So sound waves was a new technology that was not open to the public. But let's look at Life magazine here, 1965. Profound and astonishing biological revolution. Audacious experiments promised decades of added life. Super babies with inspired minds and bodies, blah, 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 blah. All of the things that you are hearing 50 years later are still in the cultural imaginary, particularly with cloning or stem cell research. I did a series of pieces about the embryo. I am very intrigued by embryos since they are marvels. They are just, perhaps, there's no way to get really inside of that concept. Those of you who have had pregnant partners may have seen some of the sonograms, and they are truly amazing. But My interest here is to kind of look at this uncharted sensibility, particularly with the patenting of life. And this piece, called Origins and Futures, in a show called Future Natural, takes one of the ideas from A.G. Carn Smith, in which he talks about this process of genetic takeover as the beginning of life. He said that primitive RNA learned how to copy itself by entering into the crystal lattice system of a particular mineral. And from that shed the matrices of the mineral itself and just learned how to copy. Now, whether or not this is true or not, and it probably isn't, because there are many competing theories. For the artist, it doesn't matter, because the idea behind this piece is to juxtapose the embryo with one of the minerals that was thought to be the matrix for primitive RNA, which is pyrite, which is called fool's gold. So the question here is, are we selling our origins in these speculative futures for now being able to license or patent DNA? And you can see a close-up, and even the bone marrow has a fetal image, and these are all rapid prototype sculptures. Several larger scale ones were maybe about 3 by 4 feet. And this one in particular that I like, it sort of starts with the Cubist baby, almost an homage to Picasso, and then with a click of a wrist and the manipulation of some zeros and ones, I can turn it into the Buddhist baby. All right, so be it. What about what's happening in the lab? And in 1986, what we saw with geneticist David Au was that he crossed a tobacco plant with firefly genes. Now, this really happened, okay? This was not a Photoshop kind of event. This really happened. In fact, when I heard about this, I called him up. He was in California at the time, to ask him why he wants to do something like this. I said, are you trying to make cigarettes that glow in the dark? And he said, absolutely not. This is how we kind of learn about how disease enters into different organisms. First, he didn't want to talk to me because he thought I was a reporter, and I told him I wasn't, so it was pretty interesting. But what you see on the right, which is from 2009, is that Japanese researchers have genetically engineered monkeys whose hair, roots, skin, and blood glow green under a special light, and who have passed on their traits to their offspring. The first time this has been achieved in a primate. And what is important in this experiment is that green glowing monkeys have green glowing babies. So if you now start manipulating the gonads or sex cells in human beings, then it becomes almost a mechanistic scenario in which you can create more of the same. I then came across a story by Kingsley, who was a friend of Darwin and wrote this very important fairy tale. And it was called Water Babies. And in this parable, he essentially, which was supposed to be read out to children in the 19th century, he has his heroine, who is Tom, who is a kind of chimney sweep, very uncouth and very rude. And as he was trying to run away from his master, he falls into a creek. And in this creek, he meets up with the fairies, who transform him into a water baby. So he develops a set of gills, and as he learns through reason and judgment to accomplish the tasks before him, he changes once again. So each time he performs his cerebral tasks, he moves up the phylogenetic order, from fish to amphibian to mammal. And this was an effort to reconcile theology with evolution. And in 2004, when I visited the museum, the Vrolijk Museum in Amsterdam, which is dedicated to all of these fetal specimens that date back even to the 18th century, I was so overwhelmed with what I had seen there, and sort of wondered what I was looking at. I was so overwhelmed, like I couldn't really even adjust to what my feelings were. I just wanted to make sure that I did not represent them in any horrible or monstrous way. And you can see in these photographs that 19th century methods were still being used as a very early technique in preservation. And in this one, you can kind of see in these triplets that the preparator was very keenly aware of, say, contrapposto, as she is setting up this scene so as to create an aesthetic appearance. So although these medical specimens may not have that much of a scientific meaning anymore, they certainly have cultural meaning, and that's the double identity of a scientific image. And here you can see part of this hit and run talk. Another kind of way to think about water babies and this kind of reconciliation with genetic engineering is back to Tom and one of the fairies, Mother Carey. And when he encounters Mother Carey, who's the chief fairy, Tom asks, he says, "I hear you're very busy." And she says, "I'm never more busy than I am now," she said without stirring a finger. "So, I heard, ma'am, that you were always making new beasts out of old." "So people fancied," she said, "but I am not going to trouble myself to make things, my little dear. I sit here and make them make themselves." So ironically, this little fairy tale in the 19th century resonates with Mother Carey's words in the sense that glowing green monkeys make replicas of themselves in the same way that Mother Carey talks about, "I sit here and make them make themselves." The Glass Veil was another exhibition, this time in Berlin, in which I was given sort of the full cooperation of the Medical Museum to create these different specimens. None of this is photoshopped either. And here you can see Shelley's Dream, which is in the basement of which was once a chapel in the hospital. You can see here that the space itself is bombed out. It's the former meeting hall of the scientists from the hospital, and the museum is very much intact in the outer rooms, but when the commission decided what to do with this meeting hall, they decided that they would keep it as is. So it is called the Charité Ruin, and that's where the exhibition was, and that's where many conferences are held as a way to kind of preserve the past. You can see the scale of this, the 30-foot ceilings. And to sum up here on a positive note, we'll look at astroculture, which is a technique that is now being used by NASA in their explorations of space to grow vegetables on other planets. And last year, a Japanese astronaut was sent out to the Mir station to grow cucumbers in space. And what he found out was that rather than having the cucumber seed create one sprout, it created two. So whereas we think that we know about the code inherent in seeds, under varying conditions and different kinds of gravitational forces, there's a whole new ball game that is out there. And I did this piece at Exit Art, where I did grow the string beans and other vegetables, and like the Japanese astronaut, I didn't eat them either, okay? So this was what the actual pictures look like. It looked like using liquid light. In a way, this fuchsia is the new green because the red and blue lights combine to create the proper usable part of the light spectrum to create chlorophyll, even though it doesn't look like chlorophyll. Back to Cara Phyllis and her idea of a biofact. In fact, we don't know by looking anymore. And here are some more of these images. And then finally, I would just like to introduce you to organ printing, another new technology in which perhaps this is your repair kit.
My name is Gabor Forgács. In the future, we'll be able to harness nature's ability to form organs and build our own.
Dr. Forgács and his lab are pioneering a radical way of building new internal organs. They're creating the organs cell by cell, literally printing copies of the original organs nature gave us.
Organ printing sounds really like the ultimate science fiction.
This technology is considered the future of tissue engineering. It's based on the fact that a cluster of human cells, what Dr. Forgács calls bioink, behaves just like a liquid. When placed next to one another, the clumps will flow together and fuse, forming layers or other shapes.
These are the bioink particles. Each of them is composed of about 10 to 30,000 cells.
To build 3D structures like organs, Forgács and his team use a printer to assemble the human cells into the desired shape. It's similar to how an inkjet printer produces words. Cells survive the printing process well.
This is the bioprinter.
Your printer can't assemble solid objects, let alone a human organ. But this one will. The printer creates a sheet of cell-friendly biopaper. Then it prints out the living cell clusters onto the paper, drop by drop. Once in place, nature takes over. The cell clusters fuse to each other, creating more complex tissue structures.
We cannot control every detail of the process, but it turns out that this is not necessary because the biological material, by its fundamental nature, is able to self-organize.
As each layer is completed, another layer of cell clusters is added.
We build the first story, then we build the second story, and we build the third story. And that's how we get to the skyscraper.
Eventually, the biopaper separating the layers dissolves, and the individual layers of cells fuse together.
And that's the way we build three-dimensional structures.
Since cells from any organ can be put through this printing process, Forgach foresees using this technique to replace any damaged organ. And since the organ would be printed from your cells, there would be little chance of rejection.
Imagine someone who had bypass surgery. That person is living with a heart which is not fully functional. So I can envision printing sheets of cardiac tissue we can attach to the damaged heart. Will I be able to replace my liver because I'm a heavy drinker? Will I be able to replace my lungs because I'm a heavy smoker? Well, my first advice is don't be a heavy smoker. But having said that, of course, it is quite possible this technology will be useful within one's lifetime. Why can't we imagine that at some point when you need to replace a damaged organ, you are rolled out of the operating theater with the new organ that was printed into your body? I think that what was science fiction 50 years ago is close to reality today.
Thank you.
Check.
Hi, Suzanne. I have a question about, I guess it's a question about artistic technique, because your physical objects, I think, are very precise, very beautiful, and then your photographs seem to have this almost point-and-shoot aesthetic, so that some of them are blurry, some of them are just like ad hoc framed. I was wondering if you could just speak about that. I guess, is my
That's a
question.
very, very good point. The photographs for me are really more about graphic symbols as well as memory devices. So whereas photography kind of prides itself in sharp focus and attention to detail, really when you think about what the other ways in which the lens can be used to evoke other kinds of emotions, I think that the blur, as in the case of Gerhard Richter, kind of creates a sense of contradiction. And I think that for the scientist or for the photographer who would essentially go into a museum to photograph some of the specimens, the idea of objectivity is what is on his mind, whereas I am much more interested in the somatic gaze in my photographs, which appear to essentially reframe or distort or create other kinds of moods that would not be clinical. And because photographs don't have the same kind of body that sculpture does, one is able to kind of create this more graphic sense, in my mind, by using the full range of photographic possibilities, including out of focus.
So I noticed a similar, also some kind of contraposition. At various points, you insisted this is not Photoshop. This happened really, or is really there. On the other side, you seem to be really interested in these kinds of science which seem to be really happening, but it sort of borders to the, I don't know, they're very much artificial. So, so this tension between the real and the artificial seems to play a role. So sometimes you insist on the distinction, but sometimes you like to sort of show how it blends into each other. Can you say something to that?
Yes,
It's
I
not a real
think
question.
that's a very good question. And as I said early on, that that artworks are essentially signs in action, and that sometimes it's really difficult to tell when you're looking at a picture what it is, what the context is, whether or not it's been manipulated or not. So there's a certain kind of distrust in pictures as well. My interest in saying that this is not manipulated has to do with the ubiquitousness of Photoshop to create these fictions, and in particularly the water babies, which were actually real specimens. When FedEx would come to pick up a package in my loft in New York and they would see some of these on the wall, they wanted to know whether these were real or not. And I said they are, and they would run out of there as soon as possible. But the interesting thing also about some of the water babies pieces, which are out of focus, this was a hit and run kind of photo shoot. Because number one, I didn't know whether I was allowed to take pictures. No one was there. I was by myself. The entire room, perhaps this big, was filled with these specimens. And I wanted to, I ended up showing my own anxiety about how I'm going to look at these things. And it took me a number of years before I even showed them. The second place they showed was more recently at the Frist Art Center in Tennessee in an exhibition called Genetic Engineering and Fairy Tales. And quite nice catalog from that exhibition. And I'm the only one who had a piece that wasn't a fairy tale. So this line, again, between the artificial and the actual gets fuzzier and fuzzier to determine. And you know, if for example, the astroculture photos were photoshopped, in my mind, they wouldn't be quite as penetrating. And sort of seeing that piece in reality and then seeing the photographs of it as liquid light, I couldn't photograph it in any other way to get that emotion. So you have these broad kinds of colors of flowing pigment that have to do with turning the camera in many ways against itself. So yes, this continuum between the artificial and the natural, between the precise and the blurry, I think all of these are tropes. All of these are ideas in which you kind of choose certain formats to express the kind of emotion that you're trying to transfer to the viewer themselves. The clinical pieces of water babies you would have just went, ugh, you know, you wouldn't want to see it. So, yes, does that answer your question?
Hi, really beautiful work. I'm curious about how you frame the objects, the sculptures that you make. The examples that you showed had a natural object next to a prototyped object. I was wondering if you feel like there always has to be that tension between the two forms of production to get across that dividing line, or could you put one in by itself and would it function?
That's another very good question, because I'm very interested in creating beautiful objects, and even though some of the subject matter that they underscore are not so beautiful, I like to use beauty as a foil. in which the viewer, in certain sense, is enamored to come and look a little bit closer. And by creating this sort of nature-culture access, objects which may have been termed too crafty all of a sudden fall into another category. That the sponges in particular, which are really beautiful objects, I think in and of themselves you would look at them more in the sense of being a ceramic than as a sculpture itself. So I think they need to be paired as much as possible in order to drive home the point.
I have a question about, I can project it on you. I have a question about your double images that you had where you would superimpose one of a natural object over a laboratory of some kind. How did you do that? Did you do it with the photographic enlarger or digitally?
Now that is photoshopped, okay? and that is photoshopped with two photographs, one which is not transparent and the other which is transparent, a Photoshop filter. And what's interesting about that process is you never know what you're going to get. So there's a great deal of chance or accident in how they're going to line up and what's going to essentially surface. And I think that that set of photographs again turned the objective laboratory into some other kind of narrative. What I did learn by visiting all these laboratories is most of them are the same. That they have the same colored gloves, they have the same colored mixers, they have the same glassware. It's almost as if this becomes a universal kind of archetype. Whereas when you visit an artist studio, they're all different.
Would you repeat the same project again with like film, black and white per se, where you could throw it on the margin and then sandwich the negatives? Would you do the same thing again?
If the images required that, yes. In fact, I did find a bunch of 2.5 by 2.5 inch, or 2.25 inch glass slides, many of which were originally used to teach art history, and I have that collection for about 15 years. Never tried to do anything with that yet either. But yeah, I mean, I think that this idea of double exposure, superimposition, this all comes out of cameraless photography, particularly during the 1920s and 30s, in which chance and accident and serendipity play a role. In fact, L'amour fou, which is Jane Livingston and Rosalind Krauss's book about surrealist photography, I think underscores this point that even though you work with a machine, doesn't mean that the outcome is predictable.
Thank you.
Thank you.